Hi, my name is Ryan Henry. Today is April 30th, 2026, and this is my DNA extraction lab. So to start, DNA is the genetic material that is found in living organisms, and it helps store the instructions cells use to grow, function, reproduce, and make proteins. So DNA is pretty important. I went ahead and did step one using the mortar method. So I soaked my peas in distilled water last night. And then I drained the liquid through the cheesecloth into the cup. And why do we add salt to this? Salt helps neutralize the negative charge on DNA, and this lets the DNA clump together more easily to separate from the solution. So now I'm adding half of the dropper full of detergent liquid to the liquid left over from the peas. Now that I've added half of the detergent, I am mixing with my glass stir rod. After this, I'm going to place it in the refrigerator for 10 minutes. So my mixture is now in the fridge for 10 minutes, and I added the detergent to break down the cell membrane and the nuclear membrane. This helps release the DNA from inside the cells. So once it's done, I'll be back. I transferred about a third of my mixture into a test tube, and then I added about half of the meat tenderizer. And now I'm making sure to mix slowly and carefully as to not break up the DNA. We add the meat tenderizer because it contains enzymes that help break down the proteins. This helps remove the proteins that are attached to the DNA, and this makes the DNA just a little bit easier to extract. So I'll be back for the next step. So I got my ice cold alcohol here, and this causes the DNA to precipitate out of the solution, and the DNA also isn't soluble in alcohol, so it's gonna kind of float on top like a stringy material, kind of obvious DNA compared to just mixed in. So I'm gonna fill this up till it's about 2/3 full. So about there. Could have held that at a 45-degree angle a little bit better, but it should be okay. Okay, I'll be back for the next step, but you can see it kind of forming up there. Okay, I've let it sit for a minute or two here, and you can see the DNA floating in between the alcohol here on top and then the original mixture down here. It's white. It's a lot more than I thought there would be. I don't know if this is the normal amount or not, but it's white, stringy. There's some suspended inside the alcohol still, and that's probably because I poured not at a 45-degree angle. If I had done that correctly, then there would probably be more settled down here. But the longer it sits, it seems like the more it kind of drops down. But overall, it's pretty cool.